So, thank you for having me here. And as Ron mentioned, I'm at the Dana-Farber Cancer Institute and the Bing Center for Waldenstrom's Research. And we're a fairly big group, and I'm going to be talking about a lot of people and a lot of things. And honestly, we all work together as a team, and we all sort of play particular roles. So Steve Trion is the director for the program, and it's sort of administrated by Chris Patterson. Jorge Castillo is the lead clinician and runs most of the clinical trials. And then where I fit in is I do all of the genetics, the bioinformatics, the sort of big data analysis, really with the idea of trying to discover new connections and new ideas. And then Guangyang takes data coming from all of us and then miraculously figures out a way to come up with new therapies and new mechanisms. And we'll talk a little bit about some of his amazing work as well. But that's the guy, by the way, who's actively developing new therapeutics, is this guy right here. He's really something else. So if you ever get a chance to talk with him, he's really, really spectacular. So before we go any further, just a couple of quick overviews. And this is really the structure of the talk. We're going to do some overviews about cancer, some brief overviews about genetics and biology, but not too much. And then we're going to sort of transition over to focusing on Waldenstroms and how these things play out. So, cancer is caused by mutations in the DNA, and the DNA is just it's the material, the chemical material that you inherit from your parents, and that sort of creates this sort of familial relationships, and why you look like your parents and why people from certain parts of the world look like each other is things are just passing from one generation to the next through the DNA. Now, it's not a perfect process, and mistakes happen. Actually, they happen more than you might imagine. And most of them can be repaired, and even the ones that can't be repaired turn out not to matter that much, and that's kind of by design. But as we get older, and as we get exposed to things like UV light or toxic chemicals, there are more and more of these mutations that accumulate in our body and in each individual cell. And that sometimes causes the function of the DNA to change. And you hear the term genes thrown around a lot in the news media, and all that means is a section of DNA that is being used for some kind of regulatory purpose. So it's going to make a protein, it's going to regulate another gene. It's just a section of the DNA that's being used as a part of the programming. And as mutations hit these genes or hit the things that modulate these genes, they change a little bit. They turn on when they're not supposed to turn on, or they don't turn on at all, or they now make a product that's a little bit malformed, and that causes problems downstream. And at some point, all these problems get to be too much, and it just overwhelms the cell. And again, 99.9% of the time, this will actually cause the cells to die, and that's it. But there is this very, very rare case where actually the cells instead start to multiply uncontrollably. And when that happens, that's cancer. So one of the things we track really closely is what tissue these things initially came out of. And that's important because it's important to realize that cancer is actually derived from your cells. It is part of you that's kind of gone a little bit wrong, grown a little out of control. And so there's a part of that cancer that still thinks it belongs to whatever tissue it is. So in Waldenstrom's, this is it's coming out of a B cell. And these are cells that are part of your immune system that make antibodies. And when you think about your immune system, it's there to fight infections. And think about the symptoms you feel when you're in Waldenstrom's, and a lot of these should feel very similar to things like when you have a really bad flu, right? Sweats, exhaustion. It's normally your antibody levels don't get quite that high when you're fighting off something, but that is actually a normal response to an invading infection. And so depending on where it comes from, it's growing out of control, but it retains some of those initial characteristics, and that can help us both understand how it's working, how the cancer is evolving, but also it also gives us some ideas of what its weaknesses might be. It may retain some of those dependencies and weaknesses from its cell of origin. And that's one thing that everyone's been looking at very closely in Waldenstrom's, is trying to understand exactly what the cells think that they are. And if we can use that information to better understand what's going on. So why we do all this, why we study genetics, why we try to study where the cancers are and what types they are, is because we've been using chemotherapy for years. And as we understood more about cancer, we come up with better therapies, right? More targeted therapies. I know a number of you are probably familiar with things like Ibrutinib or Bortezomib, also known as Velcade, rituximab. These are all drugs that are actually, not classical chemotherapies and are targeting some pathway that's being used by the cancer cell that your healthy cells can resist at least a little bit. And this allows us to put a lot of pressure on the cancer cell and allowing your healthy cells to survive. But not everyone responds to these. And, they're not always the most pleasant. And sometimes they have real lasting consequences to them. And so one of our goals is to try to understand, why is it that some people respond well to certain therapies and not others? And can we predict this in advance? And when people do progress, why are they progressing? This allows us to come up with new therapies. This allows us to better optimize things. And this is a lot of what the work that I do and that we do at the Bing Center. And actually this project that was just funded by WMFC is in fact a very large project in collaboration with the Harvard School of Public Health, Memorial Sloan Kettering, and a number of other groups actually, to really try to identify these groups and come up with ways of bringing it into the clinic, so we can so physician anywhere without all this fancy sequencing technology can figure out which therapy is really going to be the most applicable to you. So that's the goal. We're not there yet, but we're getting there. We're making a lot of progress. And a lot of this talk is going to be about how we are making progress in that direction. But before we do that, I do want to do a quick overview of genetics. And this is just simply because, all joking aside, you'd be surprised how much we forget about some of this stuff. It's fairly complicated and doesn't come up in typical dinner time conversation. And so you forget, and it's good to review a little. So the first thing I just want to get out there is, people get a little worried when you talk about genetics, because there was this period of time, especially I think in the 1980s, where there's just this sense that genetics controlled everything. I mean, I remember seeing news reports being like, there's a gene that controls whether or not you like chocolate, and there's a gene that controls this and that, and twin studies talking about how they use the same toothpaste. And it turns out a lot of that just isn't real. When you take two people and you ask a million different questions about them, you'll find stuff in common. It actually doesn't matter which two people you pick. Though there is obviously a lot of commonalities. And the way to really understand DNA is, it's not who you are. It doesn't determine what you do. It doesn't even determine what you like. It's more like a rough set of architectural drawings, right? With no design finishes in them at all. That's what DNA is, right? It's instructions on how to build your house. It's not how you decorate it. It's not how you paint it. And it's going to keep changing as you get older and you learn more things, and you realize you need an addition over here, or the color that you thought was going to be so wonderful, once it was actually up, you realize was really probably ill-advised. You can fix it, right? And this is actually how your body works too. This is how you evolve, right? This is how we all learn and grow. DNA doesn't do any of that, right? DNA is just the basic blueprint for your house. And so you are much, much more than that. And so when we talk about DNA and we talk about inheritance, really don't feel worried or limited in any particular way. So the human genome is quite big. It consists of 23 strands of DNA called chromosomes, and it's made up of 4 different building blocks. And actually, that's it. There's just these 4 different chemicals. They're strung together in 23 long strands. There are 3 billion base pairs in all those strands. And if that wasn't enough, you actually get a full copy from each parent. So you get 3 billion from mom, and you get 3 billion from dad. Which is pretty crazy. I mean, so this is a lot of information. And it's also a lot of redundancy. And this is a really important thing about biology is, you want this to be stable through generation to generation to generation. So the chemical structure is incredibly stable. It has a lot of built-in redundancy to it. It's a really impressive molecule. I definitely recommend reading a little bit about it. It's pretty interesting. But the big thing to understand is that it is big. It is 3 billion base pairs big. If you actually were to stretch the end to end, you could, it's actually quite long. And here's actually the print version in the UK that they printed out, just to kind of give you a sense of what 3 billion base pairs would look like in print. Now, this isn't just randomly floating around your cell. It's actually all housed in the special compartment called the nucleus in the center of the cell. So that location makes it even crazier if you think about it, right? Cells are pretty small, like you can't see them. And so you're talking about a subcompartment of that having 6 billion base pairs all tightly wound up. You're talking about a number of inches of DNA all wound up so tight and smushed into the cell. And so this is not only impressive from a packing and chemical perspective, it has real consequences. And it also explains a lot of things, right? Because your DNA, you inherited from your parents. So it's the same DNA, at least in theory, right? In your skin, right? In your heart, in your blood. All of that's the same. Your stomach, your muscles, all of that, kidneys, all of it has the same DNA. And yet it behaves wildly differently. And this is actually part and parcel of this exact thing. It's wound up so tightly that actually you can't unwind the whole thing, or at least it's very difficult to. There's not enough room. So in any given cell, only part of it has been unwound. And this is, the study of this is called epigenetics, because the genetics is the same. And so this is sort of like above the genetics. It's additional regulation of how the DNA itself is being physically stored and chemically annotated. And one way to think about it is if it's all so wound up, if a particular section of DNA is so wound up, it can't be seen. It's effectively not there. So the reason why your heart might behave so differently than, let's say, like your eyes, is that your heart probably doesn't see all the genes associated with the rods and cones that help you see in your eyes. And likewise, your eyes don't need any of these like really powerful striated cardiac muscle. That's not what they're there for, right? So they don't see those genes. So each cell only has a little bit opened up. And that helps determine how they respond to their environment. Now, the problem is with cancer is that this sort of organization can get degraded. And so genes that were supposed to be closed and unavailable are now open, and things that were open are now closed. And so a very classic thing you'll see, for instance, is you'll see a gene that does something that causes a cell to grow and divide. And not only does it cause it to grow and divide, it also directly causes the activation of a gene that gets transcribed and shuts that gene down, right? So it's a self loop. It's a self sort of closed loop. It gets turned on, but by turning itself on, it also activates the gene that will turn it off. So it doesn't go out of control. And then what will happen is cancer is inexplicably that gene that's supposed to come back and turn it off gets wrapped up really tightly, now it can't be seen. And as much as they try to activate it, it can't be activated. And now you don't have that off switch anymore. It just keeps on going. So that's the kind of thing that we see in cancer. And this is, again, something called epigenomics. And it's something that's pretty exciting that we're all working on right now in Waldenstrom's. And so when we put all these different things together, we didn't talk too much about RNA. And RNA is just sort of it's a very important, but it's a transitory state. And the way you can think about it is, your DNA is like your master blueprint. And it carries all the instructions on how to do everything. But you don't want to carry your master blueprint around to every single part of the work site. No, you make a little relevant copy of what it is you need. You send that part out to all the different work sites, and then they do their job and then report back in. And that's what RNA is. It's just a small copy of a section of the DNA that encodes for a gene. And then those instructions get sent on out into the rest of the cell for processing. And that will in turn make protein, and protein does most of the work, but it will also come back and interact with the DNA. It causes it to open and close, activate or inactivate a gene, which then kind of, you can see this whole loop here. That changes the RNA, which changes the protein, so on and so forth. And the most important thing about this is that this is all taking place not in isolation, but in the context of an environment, right? For a cell, it's all the cells in its immediate neighborhood. For all the cells in its immediate neighborhood, it's really the whole organ that it's a part of, let's say a kidney, right? For your kidney, of course, it's the whole organ system, right? And then ultimately your whole body, right? And then your body and how it's interacting with its environment. You just ate a tuna fish sandwich. Your whole body has to respond to that, right? You can't have it on rails where you're always going to do the same thing all the time. Your body has to be able to respond, and this is how it happens. The protein sense stuff, binds to the DNA, activates RNA, RNA makes protein, again modifying the DNA as everything changes. So this very dynamic, powerful process. And one of the things that I use as an example a lot is computers. Because if you want to think about identical clones, let's say you have a Windows PC. All Windows PCs are identical clones of each other, right? They all have the same, well, anyone who has the most recent version of the Microsoft operating system, right? Their computer's DNA is essentially that operating system. Doesn't determine what you do with it, doesn't determine how you use it, right? But it does provide that basic architecture. And none of you use your computers in the same way, even though they are all identical clones. And in fact, you see this with twins. When identical twins are young, they actually look very, very similar to each other. And as they get older, they actually start to diverge quite a bit, both in terms of health outcomes, in terms of how they appear, their personalities. And this is just part of this constant interaction with the environment and responding. And as people respond differently, make different decisions, there's sort of this increasingly bifurcating path. So I want to switch gears now. We've talked a little bit about genetics, we've talked a little bit about epigenetics, and we'll talk about Waldenstrom. How does this actually play out in the context of Waldenstrom? So the most famous spelling mistake in Waldenstrom's is this mutation in the gene MYD88. And this is a really important gene. As I mentioned, the kind of cell that Waldenstrom's is coming from is something called a B cell. And this is a part of your immune system that controls something called acquired immunity. So this is why when you get a vaccine, for instance, these are the cells that are helping actually make these vaccines work. The vaccines activate these cells, they learn how to respond to the proteins in the vaccine, and then their response to that is actually what protects you in the future. So these are really useful things, but they're all part of this like response to invading pathogens. And unfortunately, this process takes a long time to work. And so you need two systems. You need this like long-term system where you learn how to fight this thing in particular, and you need a short-term system, which is like, hey, look, we're being invaded by a bacteria or a virus, we need to do something about it while we figure out exactly what it is. And so that's called your innate immunity. And an innate immunity responds very quickly. I mean, honestly, within seconds. but it has no idea what the response is, like what it's responding to. It's just like, hey, there's something there. We need to get on this and start attacking it. And the goal here again is to buy time for your B cells and your T cells to learn how to fight whatever is coming in. And that's actually where MYD88 comes in. MYD88 is part of this quick response, innate, or I like to refer to it as dumb immune system. You know, the other one's kind of smart, figures out how to target things and go after it. This one's just sort of like, hey, this looks like a virus. I don't know what it is, but let's just kill it. And what happens is these different sensors on the top of the cell, and if they see anything that might be a virus or might be a bacteria, these sensors trigger, and they send up a little flare. And that flare is mediated by MYD88. So what happens is, like, when MYD88 is bound to one of these receptors that's activated, 2 copies of it come together. And the fancy term for this is dimerization. You might hear about that. That just means that 2 different copies of MYD88 come together. That's all it means. And when that happens, there's a conformational change. And that conformational change of the 2 proteins bound together allows for this massive assembly of other proteins to start coming onto it. And it sort of acts as a central signaling complex called a myddosome. And you don't need to remember any of this. It's not, these details are not that important. What is important is what it does, right? So this kicks off the inflammatory process, right? One cell has seen something that's not supposed to be there, and it needs to get the signal out to everybody. And it needs to get itself prepared to fight off some kind of infection. And so when you have the flu and you have a fever and you feel tired and you feel gross, that's actually inflammation. That is your part of the consequences of this kind of inflammation triggered by things like MYD88. And so it signals downstream through this gene called NF-kappa B, which is this complex right here. And as a matter of fact, when we look at Waldenstrom cells, you can actually see this green labeled NF-kappa B there inside the nucleus bound to the DNA. And when we inhibit MYD88 correctly, something unfortunately that we can only do in a test tube, all this NF-kappa B gets kicked back off the DNA and is hanging out in the nucleus waiting to signal. And most importantly, they start to die, right? So this whole process of Waldenstrom's inflammation is being mediated, can't speak today, by MYD88, and by activating this gene complex called NF-kappa B, or nuclear factor kappa B. And this is, you know, probably about 95% of Waldenstrom cases, this is what's going on. Now, Guangyang, I mentioned it before, has really taken this discovery and run with it. And he's actually been able to figure out a lot of the details about what's going on. The classical signal when we first started working in this was that MYD88 signaled through the Iraq complex. And it does. It definitely does. And in your healthy cells, this complex is much more important. It turns out in Waldenstrom's, though, it's actually a fairly minor player. It's an important player, but it's minor. The real key one is another gene called BTK, or Bruton's tyrosine kinase, that also binds to MYD88 only when it's activated. And so this activated MYD88 allows BTK to come in, and it also kicks off this process to NF-kappa B. Now, this is important because BTK is actually something that we know how to target. We actually have drugs for this, and some of you are familiar with it. Things like Ibrutinib, Zanubrutinib, Acalabrutinib. These are all BTK inhibitors that are actually now approved and being used in WM to great effect. And this is why they're so important, right? If this is the founding mutation, if this is really the thing that's driving the disease, the closer we can get to it, the closer we can get to targeting it and cutting off that signal, the better off we're going to be. But Guang didn't stop there. He actually kept on going. So he noticed 2 things. One is that NF-kappa B was upregulating these sort of inflammatory signals to the microenvironment, which is what it's supposed to do. This is known. And those signals actually get amplified by helper cells that are around in the bone marrow, in the lymph nodes. But what he realized was that this signal, in this case, he's showing IL-6, was coming back, binding to the WM cells themselves, and activating a gene called HCK, which is also being upregulated by NF-kappa B. So there's more HCK than we would expect, and now it's being activated by this secreted IL-6 in the environment. And what does HCK do? Well, interestingly, HCK can activate BTK. It can also activate a bunch of pro-growth and survival signals that are really key and very important to the cell. And it turns out that this HCK complex is really what Waldenstrom is addicted to. And we'll cover this again in just a minute when we talk about Ibrutinib. But this is a very important finding. But in typical WM, what we see happening is we have MYD88 driving BTK, getting this process started, causing this inflammation, activating HCK, which reinforces the BTK loop and also signals downstream through AKT and ERK to really form this very strong pro-growth and survival signal for these cells. And that's really what keeps the Waldenstrom cells running strong. And so this is something that we're actually working very, very hard to do, is develop HCK inhibitors. We're actually getting pretty close. We have actually some toxicology studies running on some lead compounds that Guang is running, and it's pretty exciting. So I hope there will be more to announce here in a few months. So definitely keep checking in with people like Ron and Paul, and they, as I'm sure they'll hear about it when those things actually hit. Now, one thing that is brand new, that's actually pretty recent, is a relationship between a BTK, Ibrutinib, and COVID. So a lot of people were curious. You think about what is going on with COVID and what the problems are. And a lot of it has to do with inflammation to the lung. So it infects, actually, a very, 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 very small percentage of the lung cells are actually susceptible to COVID. But what happens is once they're there, they're infected, your immune system picks up on this, and it sends an out-of-control signal to your lungs. And they just attack and attack and attack, and everybody in the general area is getting hurt. And there's a number of problems with this. It's not that it isn't good to clear infections, but it turns out that your lungs are really important for breathing. And if you have a big, thick layer of stuff coating your lungs, the oxygen can't get through. So as all these cells are rushing in to try to fight an infection, right, you can't breathe. And your lung is getting damaged, which then further increases the inflammation, which causes more cells to come in, and it creates this loop. And this is actually what gets people into trouble. Ultimately, it creates something called a cytokine storm, with all these inflammatory cytokines going everywhere, and people feel horrible. But let's stop and think about that. Wait a minute, out of control cytokines, right? Lots of feelings of inflammation, cells tracking to places they're not supposed to be, like the lung or let's say the bone marrow. This sounds a little bit like there's some similarities here, and it turns out it is. It's actually the same pathway that's being used both in COVID and in Waldenstrom. And so Steve had the idea to go back and do a review and check out and see how people who got COVID and were on BTK inhibitors or were planning to be put on BTK inhibitors fared, and it turns out they did great. Now, this is a small study, but it was enough actually to win FDA approval for a randomized clinical trial in Boston, both for Ibrutinib and for Ibrutinib. And it's actually going on right now. So, it's one of the sort of interesting things about all this work is like sometimes you study something for Waldenstrom's and it turns out you can help out with a pandemic as well. And Steve really deserves an immense amount of credit for this, not just for seeing it, but then one of the things I love about Steve is he's the guy who thinks of it, but then doesn't just be like, someone ought to do something about that. He's the guy who calls the FDA and says, we're going to do something about this. And so we did. And so hopefully here in a couple of months, we'll start getting some good results about how these actually fare. Now, it doesn't prevent COVID, to be clear, but we believe it will prevent real lung damage and hopefully hospitalization. That's really what we're hoping for. Now, one of the things about MYD88 in Waldenstrom's is almost always the same thing. And actually almost all the tests come, focused just narrowly on this one place. It's called L265P, which just simply means at position 265P, or 265 in MYD88, there was a leucine and now there's a proline. Those are just two different chemicals. They're being swapped out due to the mutation. And in other diseases, actually it turns out there are tons of mutations in MYD88 that lead to its activation. And so one of the open questions in Waldenstrom's is, why is it, right, that if you have a MYD88 mutation, I mean, 95% of people have one, and of those 95%, something like 99% of them are this L265P. We don't really see too many of these guys, and we don't actually know the answer to that. But there's clearly something very special about that in regards to Waldenstrom, and it's one of our active areas of research. But it is something that does happen. So there are people in this 1% who get some of these other mutations. And so here are the ones that we've been able to find through our screening over time. Not quite as many, and again, incredibly rare, but they do exist. And as a matter of fact, one of our initial people in the Ibrutinib clinical trial that led to its approval was actually one of these people. And we were really curious, like, we weren't being that careful, and we just had classified them as being normal for MYD88 because they didn't have the L265P mutation. Unlike everybody else who didn't have an MYD88 mutation, they responded beautifully, and we couldn't figure out why. And then when we sequenced the gene, it turned out there was just another mutation on it. So it does matter. And so if you do get tested for MYD88 and it comes back as normal, which in genetic parlance is a wild type, weird term, it's important to make sure that the testing actually covered some of these other mutations as well. And this is, and just talk to your doctor about it and just make sure, because it does have important clinical import. And it's just something that, again, you should be aware of. Now, Ibrutinib has worked incredibly well, but it's not curative, and it will eventually fail. And so one of the things that we're trying to figure out is, why does it fail? How does it fail? And can we do anything about it? And so this is a study that I did with a visiting fellow from Spain, actually, Cristina Jimenez, who was part of an Ash Scholar Award that I got to focus on the use of Ibrutinib in Waldenstrom's. And one of the things we were able to show was a couple interesting things. One was that we had, for the first time, homozygous deletions in 6q, and we'll revisit this whole idea of what these deletions actually are in a minute. But we also found these little tiny deletions, and I don't know if you can see them here, in this 8q region. Oh. And sometimes they were present in advance and they just grew stronger over the course of therapy. Other times they just seem to pop up de novo. But it was a really important discovery because while we've seen mutations in BTK itself in the past be associated with resistance to BTK, it only occurs about in less than half of the patients. So then the question was like, well, what happens to the other half? And it turns out that these 8q do in fact explain quite a few of them. And there's a number of really interesting genes, including DOCK2 and the trail receptors. And in fact, there were some mutations on DOCK2 that were observed in this study as well. And trail has long been on the short list for genes that would be important for this kind of event. So we're starting to get some really promising leads for the first time about what we might be able to do to help potentiate additional, better Ibrutinib therapy. Maybe we can use this information to help make it more effective, and B, help people continue to respond on Ibrutinib if it should fail. So There's a lot of stuff, very active research going on with that right now. The other event I want to talk about, and this is the other really big one in Waldenstrom's, is those mutations in CXCR4. And unlike MYD88, there are just a ton of them. There's tons. I mean, there's been over 50 or 60 of these things documented at this point. I can't keep track. There's more every day. But they all do the same thing. This is the sort of shorthand annotation for the different amino acids or pieces of protein that make up the CXCR4 gene. And these boxes are the ones that are actually being targeted. And as you can see, they're all at the end. And what's interesting, and if you look at the three-dimensional structure of it, these are two different copies of CXCR4 bound together. This part sticks through the membrane. This part is the receptor that sort of sticks out and tries to sense things. But where the mutations are is this part out here. And it's at the very end, a little bit in yellow. And what's interesting about it is it doesn't actually, it's not that important for signaling, right? The signaling, the binding, signaling's happening here. The binding outside the cell is happening here. This is just how you turn it off. It's how you grab a hold of the thing and you yank it back inside. And so what all these things are doing is that they're truncating or scrambling this so it can't be recognized. And then that prevents the cell from bringing that receptor back inside once it gets activated. And then the net effect of this is that people who have these mutations have this thing called CXCR4 sitting on their cell surface that they can't get rid of, that just signals and signals and signals and signals and signals. And we were talking a little bit about infection and inflammation. So CXCR4 is also part of this. It's something called a chemokine receptor. And that's actually how cells know where to go in the body. They follow a chemical trail. So you can think of this as being like a nose, kind of like a bloodhound for a cell. It sniffs out a chemical gradient, like, oh, the concentration's coming from this way. We got to head that way. And it'll get the cells where they need to go, whether it's a source of inflammation, like the lungs with COVID, or to the bone marrow, which is actually where B cells are supposed to go during their normal development. And so this constitutive activation of CXCR4 actually is pulling them out of their normal circulation, in case of Waldenstrom's, pulling them into the bone marrow and keeping them there. Now, it turns out that CXCR4 has a number of really important clinical implications. So patients with these CXCR4 mutations don't usually have much lymph node involvement. And depending on the kind of CXCR4 mutation you have, people can also present with very heavy amounts of bone marrow and very high levels of IgM. But most importantly, they tend to be resistant to certain therapies. And this includes Ibrutinib. So this is the results from our initial New England Journal study of Ibrutinib in relapsed refractory Waldenstrom's. And as you can see, we just didn't get as good responses with those people with these CXCR4 mutations compared with those people who had normal CXCR4. And this was really a critical thing for two reasons. One is these patients took a very long time to get their best response. So often full month or two longer than those people without a CXCR4 mutation. And this is important because in clinical practice, we know this and we know the hold on because, okay, they didn't respond as well, but they still got a pretty decent response. But if you're used to people responding in a matter of weeks, maybe even days, and you're out a month and you're not seeing much of a response, a lot of physicians would start withdrawing the drug. And in the case of CXCR4, if you just hang in there, they will eventually respond. Not as well, but they will eventually respond. They are also the first people to progress off, is what we've learned through additional follow-up. And again, we don't entirely know why. We have some good theories about it. But the good news is that we're getting ready to publish the results of a clinical trial using an agent that targets CXCR4, preventing its activation in people with CXCR4 mutations. And the good news is that by combining these two therapeutics, we could make these CXCR4 people act as if they were lacking the CXCR4 mutation in the context of Ibrutinib. They had very quick responses, very deep responses. And so we're very, very hopeful. And And unlike the clinical trial that we performed, there's some new drugs that are now coming out that that can be pills, as opposed to having to go into the clinic and getting an IV, which I know how much everybody enjoys, but we're hoping we're going to be able to switch that over to actually a pill format, and then something that people could do at home, Ibrutinib with a CXCR4 inhibitor, and hopefully this will erase this part of the effect. Hopefully not just for Ibrutinib, but for other drugs as well, where CXCR4 appears to play an effect. Now, after saying all this about CXCR4, you would expect this to be a really bad, this would just be bad news. But turns out it's not true. They're more symptomatic. We have a harder time getting them to respond to things, but they don't necessarily have a worse course of the disease. As you can see here, actually the overall survival is pretty similar between people who have MYD88 mutations, but whether or not they have a CXCR4 mutation doesn't seem to actually matter. People who do poorly are those who lack the MYD88 mutation. That group in Waldenstrom's tends to be very, very aggressive. And that's this green line right here. So, and this is something that we're really actively working on. And a big part of this is that this group, the MYD88, normal MYD88 group, happens, they often have the ability to transform into a more aggressive kind of lymphoma called diffuse large B-cell lymphoma. And that actually drives a lot of the problems experienced by this group. And they also, interestingly, do not respond well to ibrutinib therapy at all. And if you think about it, if MYD88 is driving BTK and you don't have the MYD88 mutation, inhibiting BTK is not going to help you. I'll give you a better example of that here in a minute. Because one of the things we did is we got a bunch of these patients, we sequenced them and tried to figure out what mutations they actually had, so we could understand how to better treat these people. And I don't recommend, no need to memorize any of these things, but a lot of these genes are actually genes that are very similar to those in diffuse large B-cell lymphoma. They're not the same, and they don't come in the same pattern. It's a very distinctly Waldenstrom's group. They also do have some CXCR4 mutations, which is, again, as far as we can tell, pretty typical, mostly found in Waldenstrom. So these are clearly a Waldenstrom's-like entity, but they're harboring a lot of mutations that are similar to other types of lymphomas, which is maybe why they can make this transition so quickly. The other thing that's interesting about this is if you look at the sort of BTK and NF-kappa B signaling cascade, and you look at where these mutations actually are, these little red triangles here, you can see they're all underneath BTK. So these are the removal of negative regulators, the activation of the pathway mutations, and you put it all together, and you could inhibit BTK all day in these cells, and they're not going to care. All their signaling is coming underneath BTK. So this is why we think that the vast majority of them do not respond. There was a study that combined rituximab with a BTK inhibitor that came out of Europe. That was actually a really great study. But I mean, I think that one of the things that lessons we had is that it turns out that they do respond fairly well to rituximab. And we've now done a number of studies, and it's just worth noting that this group has a very, very low response rate to BTK inhibition. There's only one last big mutation I want to talk about, and then I want to switch over to some big picture stuff. But it's been a big couple of years since I was away, and a lot's happened. And these are the 6q deletions. And this is the other big common event. And one of the things that was a surprise to us is it turns out they're connected. The CXCR4 and the 6q. And so when we talk about deletions in the DNA, it literally is that. I talked about there's 23 strands of DNA that make up the human genome. Sometimes they get mutated, but sometimes chunks literally just get deleted out for various different reasons. And this can have all sorts of consequences. And working with a visiting fellow from Italy, actually, Maria Luisa Guerrera, the two of us worked together to design some assays to test the locations and strength of the deletions across several different genes along this stretch of DNA. So this is sort of like half of a DNA strand for the one that's called chromosome 6. And we tested these different genes all the way up and down to see whether or not there was a copy both from the mom and from the dad, or if we were only able to pick up just one of them or none of them. In this case, the darker colors indicate only picking up one, the lighter colors indicate picking up two. And as you can see, there's a very, very strong signal where there's a group of people who have very strong deletions, like full copy number losses across the Waldenstrom's clone. We have people who are asymptomatic and symptomatic, that's the green and gray here, in both groups. But what we don't see is CXCR4. That's this over here in red, right? We see some small subclonal deletions here, but nothing else. We also don't see these 6q deletions, particularly in in patients who have normal MYD88. We see individual gene deletions, but we don't see these sort of large block deletions of the whole thing. And so this is kind of interesting, right? So it sort of paints this picture of like, okay, well, you either have a 6q deletion, or you have a CXCR4 deletion or mutation. You have one or the other. But we didn't think they were particularly related. We just thought that they were just two different pathways in the evolution of WM. It's also worth noting that both of these events seem to come after MYD88. So you get an MYD88 mutation, and then you either acquire a CXCR4 or you acquire a 6q. Now, in very advanced disease, sometimes we will see them both go together. But initial diagnosis, it's usually one or the other, or obviously sometimes neither one. And again, we don't really see them in this sort of MYD88 wild type group. But to sort of hammer home this point about the role of BTK in the MYD88 group, this IBTK stands for the inhibitor of BTK. And so this is a gene that inhibits BTK. And, one of the interesting things, even people who don't have a 6q deletion sometimes have deletions on IBTK, which makes sense that BTK is sort of the linchpin for signaling for this disease. Makes sense to have this kind of taken out a little bit. Now, what's interesting is you look at the MYD88 wild type, we saw no deletions on this whatsoever at all. And just again, hammering home that this group of patients really don't care about BTK at all. And so this is leading us now to figure out new ways to develop therapeutics for this population. But this isn't the really interesting thing. The interesting thing for us was when we tried to understand what was a consequence of the 6q deletions and what was a consequence of the CXCR4 mutations. And what we discovered was that they're not the same, but they're very related. As a matter of fact, there's a group of genes that are pretty much identical, right? If you compare them to patients who don't have a CXCR4 and don't have a 6q deletion, it turns out both events affect genes, as the set of genes, about the same amount and in the same direction. So if it was upregulated in one, they both bring it back down. If it was downregulated, now they're bringing it back up. And again, by about the same amount. Now, this is really fascinating. And I think one of the ways that you can think about this is, if you're familiar with the phrase, all roads lead to Rome, you can think of all mutations eventually lead to WM. You have to figure out a way to get there. And there's different ways of getting there. But ultimately, you want to wind up in the same place. And so the fact that these overlap so heavily in their signaling, despite the fact they have nothing to do with each other, really indicates that there's a set of genes that really need to change in Waldenstrom's in order for this to come up. And the ability to identify these genes and be able to target them is a really big step forward in our understanding of the evolution of Waldenstrom's and how it goes from being sort of an indolent thing that nobody cares about to being the sort of problematic cancer that we all know about. The other thing that we've learned is that, Waldenstrom cells are sort of stuck between two states of your immune system. One is called a B cell and the other one is called a plasma cell. And you can think about B cells are things that are going out and looking, helping you remember old infections or learning to fight new infections. And plasma cells are like dedicated fighters. It's like Once a B cell gets tapped, and it's like, hey, you're really good at fighting this infection. You go into the ring. It goes and turns itself into a plasma cell, and all it does is produce these sort of fighting like proteins called antibodies that will help fight off that particular infection. And Waldenstrom is interesting because it's stuck about halfway in between the two. And when we look at this, like the gene usage pattern, we can actually see sort of where they are along the spectrum of transition from a B cell to a plasma cell. And it turns out it depends very, very heavily on the mutations that they have. So people who are lacking MYD88 mutations, for instance, are much closer to B cells. People who have CXCR4 mutations and MYD88 mutations are somewhere sort of in the middle. And people who have MYD88 mutations but no CXCR4 mutation, well, they're much closer to plasma cells. Now, if you think about this, this has some implications, right? Well, if the CXCR4 people are less differentiated and the MYD88 people are more differentiated, that means that the 6q deletions are happening in this context of a more differentiated, more plasma cell-like version of Waldenstrom. So you can almost think of this as that we have two different ways of evolving Waldenstroms, one that's more plasma cell-like and one that's more B cell-like. And as we've started to study like differences between WM, understanding how Waldenstrom's is different from healthy donor B cells and how WM is different from healthy donor plasma cells, we can really start to piece these things together and understand how Waldenstrom's is being dysregulated, what's unique to Waldenstrom's, and what is just happening to do with their different locations in the sort of differentiation state. And it's also, we're getting some validation for this research from all over. A really good friend of mine, Chris Oakes from Columbus, Ohio, recently published a study looking at the epigenomics of Waldenstrom's and actually came up with the exact same thing that we did from the epigenomics standpoint, and was able to show that looking at methylation or sort of chemical annotation of the DNA, that Waldenstrom's split up into two different groups. One more B cell-like and one more plasma cell-like. And sure enough, the plasma cell-like ones had all the 6q deletions, and the B cell ones had all the CXCR4 deletions. So again, they're doing something very similar. They're activating the same set of genes, but in two different contexts. And this is now how we sort of are starting to think about this. Is from a review I actually wrote for that paper. You can think about, these are the healthy donor memory B cells, these are healthy donor plasma cells. Waldenstrom sort of exists on the spectrum, and based on these MYD88 and CXCR4 types, we can now start to talk about what kind of therapies they respond to, right? What their clinical presentation is, and what other kind of mutations and deletions often accompany them, and how these cells are developing in different ways, depending on these different variables. And we're really hoping that we can continue to build and refine this kind of modeling to be able to look at patients, evaluate them, and then be like, you know what, this therapy is the right one for you. Maybe like this is normally really great, but in your case, this is what we want you to have, and have much higher success rate and less time chasing after therapies that ultimately aren't going to work. So just a quick review here. We covered a lot of stuff, and I know I'm sure everyone's feeling a little overwhelmed at this point. I'm feeling a little overwhelmed at this point, and I made all the slides. We have MYD88 here, and it's, again, part of this quick response sensor complex. We're looking for infections. And it activates the IRAX, and it activates BTK, and activates NF-kappa B, and goes out and signals to the cells, right? And you have CXCR4 that's also signaling and is also helping the cells move through the immune system and locate themselves, in this case in the bone marrow, in the case of COVID, to the lungs. And there's mutations in them that make it constitutively activated. But the good news is we've been able to use this information to develop new therapies. So we have Ibrutinib, which is now clinically approved, venetoclax, which is targeting the cell survival mechanisms that we didn't really talk about in this talk, but we now know are dysregulated in Waldenstrom's. We just completed this clinical trial with Ulacupimab, which is unfortunately not going to be available ultimately. The company decided to go with a different direction for it. But because it was a successful clinical trial, we actually are now pursuing some of these oral inhibitors, and there's a lot of excitement from a number of the partner companies about developing these. Actually, they are developed, about doing clinical trials in Waldenstrom's in the context of BTK inhibition, CXCR4 mutation. So this is really an exciting area. And again, I hope we'll be able to give you some hard details about that in the coming months. We're also developing our RAC inhibitors. So even though these are not the majority of the signaling, it's mostly going through BTK, you know, these still signal and they're still important. And one of the things that we're trying to do is do this dual inhibition technique, and to see if we can get better responses. And actually, with the idea that if we can really cut off NF-kappa B, maybe we could actually start not just holding it steady, but actually killing the cells instead. And ultimately, the other thing that we have here are MAP and ERK inhibitors, and also HCK inhibitors, which aren't shown here. And the HCK inhibitors, I think, are maybe the ones that we're the most excited about at this point. It's one of the interesting things we've discovered about Ibrutinib resistance, for instance, is that this, it turns out that it's actually the inflammation is what Waldenstrom's is addicted to. And one of the reasons why Ibrutinib works in COVID and works in Waldenstrom's is it cuts off that inflammation. It's why people who've taken it report getting their energy back, this, you know, night sweats going away, feeling a little bit better. A lot of the sort of those type of symptoms disappearing quickly on Ibrutinib is because the inflammation is going down. And in COVID, again, it's the same thing. It's getting the body to take a step back and not invade the lungs quite so heavily. But when it gets mutated, unfortunately, it can still signal. And if you get this inflammatory signaling environment back, it doesn't matter if you have the mutation or not anymore. You're still in this sort of inflammatory environment. And apparently that's enough to make the cells thrive. And so we've seen these situations where only like 10% or less of the cells will carry a BTK mutation, but all the cells become resistant to further BTK therapy. So we're hoping that by targeting HCK, we can actually sidestep this process and cut off the cell's ability to sort of respond to the inflammatory environment, and be ultimately much more effective. But again, this is something that's in development, but we're very, very excited about. So I'd really like to thank everybody for your attention. There's a lot of information. We had a lot of updates in the last 2 years. And I'd like to thank, of course, the WMFC for their generous support, both financial, but also personal. I really appreciated you guys bringing me out there a couple of years ago and showing me around. And it was really a beautiful thing. I just like to thank you all for that. Also the IWMF, everyone at the Bing Center, we work as a team. All the data they show really belongs to everybody there. They've just done a ton. We've been working a lot with the Jerome Licker Center for Multiple Myeloma to help us put Waldenstrom's in better context. We have a SPORE Career Development Award, Nash Scholar Award, and of course a strategic roadmap grant from supported partially by IWMFC, the LLS, and of course the IWMF as well. The Orzag Family Fund, the Cody Bailey Fund, the Motto Fund, and most importantly, actually, the patients. And because honestly, like, we wouldn't be able to do any of this if it weren't for the support that we received from all of you. So thank you very much, and I'd be happy to take any questions, including some of our future research that we're just getting started, or any questions about the presentation that you might have.
So can we unshare Zach's screen, maybe?
I'm sure
there,
did it work?
sure. Done.
I will unmute people.
Okay. Perhaps, Zach, I'll go through the questions that I think Charles has written a few questions down in chat. So he's been quite excited about an ibrutinib knockoff in India called Ibrishil, and I'm wondering if you're familiar at all with if that's likely to make its appearance anytime soon in North America. I would think there would be patent laws that might keep it away for a while, but do you have any insight on that?
So I'm not familiar with Ibrishil specifically. There are a number of ibrutinib knockoffs. Some of them have been successful, some of them less so. But some of them actually look really good, to be honest. And not always better, sometimes just different. And that's not a bad thing, right? I mean, again, a lot of this is about tailoring things to the person, right? And so we're actually, yeah, we're very excited about it. I'm not familiar with Ibrishil, but I can definitely tell you that they are probably trying to get clinical trials running here. Whether or not for WM, I don't know. You know, Europe, the US, Canada, Australia represents very, very large markets. And so companies who own compounds like that, they want those clinical trials running, right? Because that's their revenue stream. So they're very interested. So I would expect that if it's successful and people are buying that it's successful in the studies that they're producing, that we will see that crop up in North America relatively soon.
Yeah, because I think current sticker price is about a tenth of the cost of ibrutinib. So I think.
Oh yeah, that is just. I can't comment on this, but like I can't believe it. Yeah.
Charles has got another one that I think is related both to Waldenstrom's and to, I would say, COVID. He says, is there a link between the strength of the cytokine storm and the strength or health of our immune systems?
That's an excellent question. You know, we don't really know is like why cytokine storms happen for some people and not for other people. You know, and I think COVID is kind of a great example. You know, the vast majority, well, not the vast majority, a bunch of people with COVID don't even know that they have it, right?
Yeah.
Which is weird. Another group of people get a little bit sick. They cough for a while, and then they're fine. And then, you know, and unfortunately in the news media, like what you hear about is like, oh, people who are older, who have pre-existing conditions, they're the people who do really poorly. And that's true, right? That is true. Those are the people who are at risk for dying. What they don't tell you is that roughly the same percentage of like 20-year-olds are also getting hospitalized and they're also going on vents. The difference is that they survive. So who gets hit hard? We don't really understand what that link is, but there's clearly something about either the genetic makeup or the epigenomic makeup that really gives, that really seems to be determining whether someone has a strong reaction. And I think that cytokine storms are part and parcel of that. We see very similar things with sepsis. I mean, it's always very serious in sepsis because sepsis is when you have an active infection in your blood. So that means it's getting all over your body. It's a very dangerous condition. And, but one of the things that will typically kill you first is actually not the disease, it's the cytokine storm. Yeah. And some people just, it's so bad and it's so strong, it'll kill you incredibly quickly. Other people will be fine. And why, we don't know. But it's an excellent question. And there's some great work actually being done at Boston University looking into that for those who are interested.
Okay, Charles had one more. Worldwide, would you have an estimate regarding percentage of research that is specifically dedicated to finding a cure for Waldenstrom as opposed to finding treatment to manage or control it?
That's a great question, Charles. I don't know how to answer that. I would say for the most part, you know, for the most part, we always are like, you know, we're always really trying to knock it out of the park. Like we would like to get the cure. And, you know, I think when we were targeting BTK, we were really hoping, the goal was not control, but control is what we got. And that's not a bad thing. We're actually really I mean, it's the most amazing therapy we've gotten for WM, and it's really turned things around, especially for people who have relapsed refractory disease. But no, I mean, if you're asking if we're satisfied about it, no, we need to do better than that. I would say that there aren't that many people who are just looking for control. But again, if we're not sure how to get the cure, or we develop something that we think is going to be a cure, but it looks like it'll actually just be a control, we're not going to just like throw that out and be like, oh, we don't need that, like cure or bust, right? No. We're happy with control if that's what we get. But I don't think that's the goal for anyone, even though that's often what we wind up with.
Okay, thanks. And a question from Paul. Would you be able to give us a quick summary of this research project that's just been announced to be funded, your 300 Waldenstrom's patients study?
Yeah, Paul, thank you. I promise I didn't pay him to put that question in there. No, I'm actually really excited about this. So we actually already have the 300 patients. We actually already have the sequencing data. So that's actually not what's being funded here. What's being funded here is the opportunity to bring a whole bunch of people together in what is maybe one of the largest and most comprehensive data sets in lymphoma in existence right now is in Waldenstrom's, thanks to the support of WMFC, or WMFC and IWF and the LLS. So the goal of this is to take all these different pieces together that were actually derived from the same bone marrow. So it's 300 untreated patients, and from single bone marrows, we were able to get some epigenomic data, some genetic data, some it's called transcriptional data, which is a measurement of how the genes are actually being used at a given point in time, and try to bring it all together, and with a couple different goals. And if you think about the CXCR4 MYD88 thing, it's kind of a good example. You have two different events. They're completely unrelated to each other, right? Their signaling pathways have no obvious intersections. They're in completely different parts of the DNA. There's nothing to link them that we would think about in biology. And yet, they do more or less the same thing in Waldenstrom's, right? We would not have been able to predict that from the genetics. But when we looked and were able to combine the epigenomics and the transcription with that data, lo and behold, there actually were a bunch of genes which were being regulated in the same way. And as a matter of fact, what we're hoping we can find is to be able to group and classify different mutational patterns into broad categories. And a way you can think about it is if there's a signaling chain, right, where you go from A to B to C to D to Waldenstrom's, right? You could activate the top of the chain, you could activate the bottom of the chain, and actually you probably won't be able to tell the difference. The chain's activated either way. Now the problem is we may not know what that chain is. And we may look at these two different events and not realize they're part of the same chain. And so it's like, hey, there's people who have this, and there's people who have that, and what do we know? Right? But when we bring lots of this stuff together, there's a lot of, we can use some advanced machine learning techniques to basically try to harmonize the mutational patterns, the epigenomic patterns, and these sort of gene usage patterns together to look for these natural groupings. And then we can use basically our biology information and skills to then sort of try to understand why they're grouping together. And so we're hoping we can come up with different subtypes of Waldenstrom's. This has been done successfully with some other cancers. And then take that information, we're not stopping there, right? Try to figure out what signaling patterns are active in each of these different groups. How are they same? How are they different? And this is using, again, some very advanced machine learning stuff from the Harvard School of Public Health. And this will help us in terms of trying to figure out therapies that might work best for each group, hopefully coming up with new targets, but also maybe explaining why certain people respond to existing therapies as well. And then the third piece of this is what I think is the most critical, is it's great that we can come up with all these fancy groupings and understand why certain people do like respond to this or that and this within the study. But we can't, in practice, have you come into the clinic, have you get your DNA sequence, get your RNA sequence, get your epigenome scanned, have someone come in, spend several weeks analyzing it to come out and be like, oh, hey, you have Waldenstrom's type B, right? That's not possible. We can't do that. I wish we could do that, but we can't do that. So what we can do, though, is we can come up with proxies, right? We can come up with tests that are unique to each group that can be run by anybody, that any pathology group anywhere in the country can go and say, oh, hey, you know what? They have Waldenstrom's. We're pretty sure about that. Here's a couple of tests we can do to figure out which type of Waldenstrom's they have. And once we have that information, this is really critical. We can go back and we can look at historical clinical trials and figure out which, like how these different groupings of Waldenstrom's played out and how they all responded to different clinical trials. We can incorporate that into prospective clinical trials, right? If there are important pieces of care and clinical management that we need to know about these groupings, again, it would be very easy for someone to come in and figure out what, like, which group a particular patient belongs to, and therefore all the information we've learned about this group can then be brought to bear on behalf of caring for that patient. So that's really our goal. Like, use some really complicated machine learning techniques to understand the grouping patterns and the signaling patterns within the groups of WM, but then also to develop these tests that allow anybody, really, any hospital anywhere, to go and quickly and easily determine which group each patient belongs to and be able to apply what we learned from these studies to care for that patient. So that's the study, that's what we're up to, and that's what you all are actually making possible.
Well, that would be fabulous if local doctors could get that kind of directed support and be able to get to quicker diagnosis and quicker determination of the best treatment plan. So good luck with that. And you're looking, what, at a couple of years to?
2 years. 2 years, yeah. I mean, luckily, is again, we actually already have the sequencing data. And so we actually basically can get started right away. So we can really hit the ground running. So we're, and actually everyone's already lined up and ready to go. And the public health people are already actually designing some of their, or customizing some of their algorithms to this data set. And the people over in Memorial Sloan Kettering in New York, who are helping out with some of the epigenomic stuff, are already doing some differential cell of origin analysis and other things for their group as well. So like, people are already getting started. And again, I think it's gonna be really great. And I don't know how many of you have tried to get MYD88 or CXCR4 testing done, but I can tell you, at least in the US, it's a mess. I mean, half the MYD88 tests that are offered like aren't very sensitive. And so one test will say yes, the next test will say no. You know, and the poor physician has to go through and figure out which one to believe. I mean, it's kind of like COVID testing, right? It's just this like big like free-for-all out there. So we're trying to come up with more reliable, easier to reproduce strategies, essentially, for doing all this.
Does anyone else have a question for Zach?
Ron, it's Paul Ferguson. Can you hear me?
Yes, Paul, please go ahead.
I do have it. So the doctor will hear the question I'm asking. We're newbies on Zoom. This is our first use of it.
Welcome.
Okay. Doctor, thank you for the presentation. It was really informative. And I noticed early in the presentation, you made mention of side effects. And I've experienced since the diagnosis what I consider to be side effects of the WM, and just would like for you to comment on it. It consists of hearing loss and challenges with my balance. Is this something that is common with other patients?
It's not uncommon. I mean, it's different for everybody, but no, that's not uncommon.
Okay.
That definitely is a thing.
Okay, so it's not unusual.
Yeah, I mean, it's not super common either, but it's not, I mean, that doesn't surprise me.
Okay.
Well, sometimes things like peripheral neuropathy can lead to balance problems as well if you have that in your feet.
Mm-hmm.
I know that I used to have better balance than I do now.
Yes, it's led to balance problems for me.
Any other questions for Zach? He's given us almost an hour and a half, so we wanted to let him get back to his family, but anyone else?
Oh, I see someone for, John wrote, Halifax won't do genetic analysis prior to Bruno, why not? That's too bad. I think that they really should. We're really encouraging people to do that. You know, we actually encourage people to get both MYD88 and CXCR4, but I think the real critical one is going to be MYD88, just because we have such a low response rate to ibrutinib for MYD88 normal individuals. It just doesn't seem like it just doesn't seem worth it to put somebody through a therapy that they're not actually going to respond to or have a very low likelihood of responding to. Now, the question is whether or not they're able to do the genetic analysis with sufficient sensitivity that they feel confident in their result. And this is a problem throughout the US as well. And so that might be what they're dealing with. And, you know, and different groups deal with it differently. Some people just power through it anyway. Again, kind of like COVID. And then like you get a negative result, but you're like, is it really negative? I'm not sure. I got really sick the other week. And yeah, I mean, unfortunately, not all tests are created equal. And sometimes people move forward with tests that are only, let's say like, you know, 80% accurate, which is good, right? But that means that you're missing a lot of people. And if you only expect the MYD88 wild type group to be like 5% or less of your population, you know, that means the vast majority of people you're calling wild type, if you're, you know, they're actually MYD88 mutant. So this has been an issue for everybody. And there's no easy answer. I mean, what would be necessary to improve the reliability is pretty expensive. And so you'll see it like Memorial Sloan Kettering, you'll see it at, you know, at Dana-Farber, for instance. But most groups, even big hospitals in the US, just, you know, they don't have a genetics focus, really just don't, you know, can't do it.
Okay, thanks, Zach. Anyone else?
I follow up on those two questions that I posed. I'm curious, if I were looking for a link between, say, IgG level and cytokine storm level, is there any areas of research that you've been into that I could follow up on if I was using a Google search?
Yeah, so they're not the same thing. They both are related to inflammation, but they're different. And so like Waldenstrom's people don't have a full cytokine storm, or I mean, cytokine storms you can't survive very long in one. So Waldenstrom's have sort of a more lower level, but chronic inflammation. That's pretty serious, but not quite the level of a storm. And I would be surprised if there wasn't a link between the two, but they are regulated differently. So it's not quite so straightforward as all that.
Okay.
So there is some interesting literature that was published by Guang again. But I don't think it's been formally published in the journal. I think it was just presented at a meeting. I can try to find that for you if you'd like, and I can send it to Paul. Does that work?
Yep, and I'll send it on to Charles.
Okay, thank you for that. The other follow-up, would you consider CAR T therapy a cure?
Oh, interesting. I mean, here's the thing about CAR T therapy. You know, I mean, it suffers from the same thing that we're talking about of all other therapies, except it's new and exciting. Like, for some people, yeah, it's a cure, right? I mean, there's some diseases we thought were completely uncurable, and it cures them. That being said, it doesn't cure many of them, right? Just some people, and we don't know why, right? Why some people, not other people. It's also a very hard therapy. It's not a walk in the park, at least again, for most people. It actually can cause something that's sort of similar to a cytokine storm. It's called, basically it's related to the fact that it can kill off so many cells so quickly that it creates something similar to a cytokine storm that can be very, very hard on somebody's body, and it can be quite dangerous actually. So you have to, especially in the case of Waldenstrom's, we have to be a little bit careful with it. It's not super well studied, but it's a very exciting thing. I think that this is, you know, it gets a lot of play in the media because it's really exciting and because there are these few cures that get published. I think it's something to keep your eye on, but I don't think it's, in the case of Waldenstrom's, I don't think it's there yet. I think it's maybe a little bit, it's not my place to say. I'm not a treating physician. I don't do anything like that. But I've yet to hear of any physician really strongly recommend anything like that. Definitely not outside of a clinical trial. Just because it's dangerous enough, and Waldenstrom's, especially in the context of Ibrutinib, is indolent enough, it's just not worth the risk. It's also the efficacy in diseases like Waldenstrom's tends to be quite low. So it cures some people, but again, the vast majority, it's like a normal therapy. It works for a while and then it doesn't work.
Thanks for that.
Thank you so
but
much.
Someday, hopefully. I mean, actually, it's really a promising thing. So keep an eye out. We'll see how it develops.
Well, thank you so much, Zach, for joining us again in the Great White North. It's just fabulous to reconnect with you. We had such a great time in Halifax, and it's great to have you here virtually. And please keep us posted on progress of your project, because that's pretty exciting to think that a roadmap might exist for really any hospital to guide us to the best therapy in the future. So thank you.
Well, thank you, Ron. No, and thank you for having me. It's a pleasure to be able to be here with all of you.
That was quite a half hour we asked you for.
Sorry, I went on a little long there. I hope it wasn't too much.
Thank you so much. You really were
Thank you, Jim.